Hi everyone, what we have here is a brief review of some of the important concepts that are related to enzymes. So first of all, in terms of terms, enzymes are catalysts that speed up the rate of chemical reactions. We know that enzymes are proteins. Enzymes commonly break apart molecules. They can break apart molecules and they could put molecules together. As an example, we have an enzyme named lactase, whose job it is to break apart lactose. So if an enzyme is working on a molecule of lactose, the lactose is also known as a substrate. So the substrate is the molecule being worked on by the enzyme. When an enzyme and substrate work together, there's a specific part of the enzyme at which the reaction takes place, and that site is called the active site. So if you look at the diagram on the bottom of this slide, we have an enzyme, that's the big gray thing, and it has a round and triangular shaped active site. You then see the green substrate fitting in very nicely in our enzyme substrate complex. And then you notice that the green substrate is orange and grayish. What that is showing you is that when the enzyme works on the substrate, the molecule of the substrate changes. And what we end up seeing here is that the substrate now changed to two products. So if this were an example of, if the enzyme were a protease, for example, the protease would break down a protein, a complex protein, into two simpler proteins. You notice on the last slide that the shape of the substrate fit the enzyme exactly. And what that represented was something known as the lock and key model of enzyme function, which tells us that enzymes are specific. Every enzyme has one specific job. An enzyme that breaks down carbohydrates can only break down carbohydrates. It cannot break down proteins or lipids. What makes an enzyme specific is its shape. Each enzyme only works with one type of substrate. And over here I have three examples: lipase, protease, amylase. These are three enzymes found in our intestines, but lipases are specific to fats, proteases are specific to protein, and amylases are specific to carbohydrates. If you look at our cartoon diagram over here, the circular shaped substrates, the red and the green, will fit into our orange enzyme, but we'll need the rectangular shaped substrates, the blue and the purple, to fit in to this enzyme. And what this cartoon is showing us again is that the specificity of the enzyme depends on its shape. Now, our enzymes in our body tend to work well in our body's internal environment. But we'll see that different environmental factors affect enzymes differently. What we see on this graph is how temperature affects the rate of enzyme reactions. So at a low temperature, the enzyme activity is not very high. As the temperature increases, so does the rate of enzyme activity. It increases until about 37 degrees Fahrenheit. That is our body temperature. And once an enzyme reaches that point, once it starts to get too hot, the enzyme activity starts to decline. So every enzyme has an optimal or best temperature at which it works. Below that temperature and above that temperature, the enzyme activity will decrease. And in our bodies, a high fever is really dangerous because high temperatures can cause enzymes to denature or to lose their shape. And once an enzyme loses its shape, it can no longer function. Another factor that affects enzyme activity is pH. Now, most of the enzymes in our body work at a neutral pH, a pH of 7, but there are some enzymes that have different requirements for the pH of their environment. Pepsin and trypsin are both enzymes from our intestine, but you'll notice pepsin works in a highly acidic environment, an environment that has a pH of about 2, whereas trypsin will work best in a slightly basic environment, an environment with a pH of 8. Another thing that can affect the rate of enzyme activity is the amount of substrate within a solution. So if you have an imaginary cell with 100 amylase molecules, if there's only 10 carbohydrates in the cell, so we know not all the enzymes are working. And that's what we see here. When there's not that much substrate, the rate of the enzyme activity is not that high. In our imaginary cell with 100 enzymes, as we add more and more substrate, more of the enzymes become used and the enzyme activity increases. So in our case of 100 enzymes, once we get to 100 substrates, all of the enzymes are working and the enzymes reach their point of saturation. At this point, the activity of the enzymes can no longer increase. So another way of explaining this graph is saying as you add substrate to solution, the rate of enzyme activity will increase until all enzymes are involved in reaction, at which point the enzyme activity levels off. And that's what this flat line means. So we just went over, and we also looked at how different things such as substrate concentration, pH, and temperature affect